So here we have a very basic science type of question, right? We have a pharmaceutical researcher is looking through this nuclear enzyme inhibitor, and it's essentially asking the question, one of these enzymes catalyzes methylation of something. What genetic process does that impact? So it's essentially asking methylation, what does that do? And secondly, how does that impact, actual human beings? How does that genetically modify any of the DNA? Now, methylation essentially means adding a CH3 group to parts of the DNA. And what that does is essentially it forces the histones to kind of bind very, very tightly. It kind of closes off that area of DNA from any transcription. On the other hand, when we have something called acetylation, it actually opens up that DNA. It allows for that DNA to be a little bit more accessible for the DNA polymerases and all of those other enzymes to get in there and actually transcribe the DNA and translate it into proteins to actually, create something out of the genetic code that exists within the DNA. So here we have multiple different option choices, right? We have aneuploidy, which is essentially the presence of the abnormal number of chromosomes, and so that isn't correct in this particular situation. Epistasis is essentially when the allele of one gene affects the phenotypic expression of alleles on another gene. And so in this case, this is not relevant. Imprinting is the correct answer here because it's essentially when one aspect of the genome is essentially inhibited from producing a specific type of gene. And so it's imprinted and be carried on through generations as well. And then we have meiotic disjunction, which is similar to aneuploidy, where we have the abnormal number of chromosomes. And then we have pleiotrophy, which is where one single gene influences multiple different phenotypic traits, kind of as opposed to epistasis. So the answer here is going to be imprinting.